The presence of acetylcholine in immune cells and its correlation with age and inflammatory diseases, by Doretta Thompson. Acetylcholine is viewed as a neurotransmitter in the cholinergic system involved with transduction of action potentials. Most innate and adaptive immune cells respond to neuronal stimuli by releasing or expressing cholinergic components to form the non-neuronal cholinergic system. These systems are involved in neuroimmune communication between the peripheral immune and central nervous system, regulating immune responses to maintain homeostasis. Dysregulation in neuroimmune communication may lead to inflammatory and autoimmune diseases. Increased age causes a decline in the antioxidant defense system, which fails to maintain redox homeostasis, leading to the activation of the nuclear factor kappa beta pro-inflammatory pathways and pro-inflammatory cytokines. Chronic inflammation is associated with chronic diseases like dementia, diabetes, heart disease, and which is more prevalent in older age. The study objective is to determine the presence of acetylcholine and expression of cholinergic genes in sorted immune cells from mice and human peripheral blood mononucleocytes and the correlation with age, gender, and inflammation. B cells, T helper cells, T cytotoxic cells, splenocytes, and bone marrow cells were sorted from pooled mice cells, and intracellular acetylcholine concentrations were quantified by mass spectrometry. Frozen human peripheral blood monocyte samples were obtained from elderly participants from a vaccine study. RNA was extracted and quantitative cyber-green PCR was performed to determine expression of cholinergic genes in immune mice cells and human PBMCs, and showed expression in most immune cells. Preliminary results on human PBMCs indicate a possible decrease in expression of CHAT in older participants. CHAT is responsible for acetylcholine synthesis. A bigger cohort study on sorted immune cells and human PBMCs will be done and will allow us to gain more information on the correlation between acetylcholine and cholinergic gene expression with age, gender, and inflammation.